Okay, let's unpack this. Welcome to the deep dive, where we take a stack of dense source material and hand you the crucial insights.
Glad to be here.
Today, we're peering into the pharmaceutical lab of the future. We're not just talking about finding drugs, we're talking about engineering the perfect keys that fit the most stubborn locks of disease.
And doing it with a whole lot of computational power,
Yeah.
some really breakthrough chemistry.
Exactly. And this is essential listening because the sources we have, they show the early drug discovery pipeline, a process that's, famously slow and expensive, is really undergoing a revolution.
It really is. We found three core advancements that are sort of converging. First, a radical new way to screen massive chemical libraries without the usual constraints.
Okay.
Second, a strategy for computationally designing brand new therapeutics for targets we just couldn't touch before.
And the third.
And third, the advanced AI that's really providing the intelligence layer for all of it.
And before we get into the tech, we need to understand the scale of the problem they're trying to fix. You, the listener, should care about this because the current methods are just so prohibitive.
They really are.
I mean, the industry standard high-throughput screening HTS, our sources call it out as costing literally billions of dollars.
Billions, and it demands this gigantic specialized infrastructure.
Right. So that cost barrier, that whole logistical burden, it means only the absolute biggest pharma companies or, the best funded academic centers can really play in that early discovery space.
Precisely. We need systems that democratize this. And while affinity selection methods were an improvement, they always ran into this massive technological wall.
A wall related to how you identify the molecules that actually bind.
Exactly. Which brings us to our first breakthrough, solving the barcode problem.
Okay, so let's start there. With the technology, a lot of discovery labs rely on DNA-encoded libraries, DELs. Can you explain what those are and then why they're so fundamentally limited?
Sure. So DELs were revolutionary. They let researchers screen hundreds of millions of compounds really quickly by attaching a unique DNA strand, a barcode, basically, to each small molecule.
And that barcode tells you what molecule bound to your target.
It's the key. It tells you exactly what you have after the selection process.
But the barcode is also its Achilles heel.
Absolutely. The source material highlights a truly staggering fact here.
Which is?
That DNA tag can be more than 50 times larger than the small molecule it's tagging.
50 times. That's not a tag. That's a small boat tied to a key.
I mean, imagine trying to use a delicate house key, but you have a giant heavy backpack strapped to it. It just can't work properly.
No wonder that was restricting the binding.
It creates huge problems. First, that giant tag physically gets in the way of the molecule finding its best binding pose.
Okay, that makes sense.
And second, it fundamentally biases the screening. So if your target protein happens to bind to nucleic acids, like a lot of transcription factors do, it might just stick to the huge DNA tag instead of the drug.
Giving you false positives or just, a mess?
A total mess.
Yeah.
And on the chemistry side, you're limited to reactions that won't destroy the DNA barcode itself.
So only water and DNA compatible reactions.
Exactly, which cuts you off from a huge swath of really useful complex chemistry. Things like cross-couplings and heterocyclizations that you need to make good, druggable molecules.
So the self-encoded libraries, or SELs, they basically said, forget the barcode, we'll ditch the DNA entirely.
That's the idea.
So how do they identify the molecule if there's no tag?
This is the genius of the SEL platform. It's a barcode-free method. They still use combinatorial synthesis, but they do it on solid phase beads.
And with no DNA, they can use all that complex organic chemistry.
Right. Then after selection, instead of reading a DNA barcode, they identify the hits using mass spectrometry.
Wait, let's pause on that because that's a critical technical nugget. Mass spectrometry, MS, that just measures the weight of a molecule, right?
Correct.
If they're screening, up to 750,000 small molecules at once, won't a lot of them have the same exact mass? You mentioned mass degeneracy.
You hit the nail on the head. That is the challenge.
Yeah.
Simple MS is not enough because you might have hundreds of isobaric compounds, different structures that weigh the same.
So they needed another layer.
They did. They had to introduce tandem mass spectrometry, or MSMS.
So for you, the listener, we're not just weighing the molecule now, we're basically blowing it up and mapping the fragments.
Yes, exactly. That fragmentation pattern becomes the molecule's unique chemical signature.
But that must generate a massive amount of data.
It does. So they developed custom software called COMET, the Combinatorial Mass Encoding Decoding Tool.
COMET.
What COMET does is it predicts what the MSMS fragmentation pattern should look like for every single one of those 750,000 structures, and then it matches the signatures it finds against those predictions.
So COMET is the sophisticated decoder. That's a huge technical leap.
It is.
And the sources gave some compelling proof, right? First against carbonic anhydrase, the nice.
CAX, yes, where they found the binders they expected to find.
But then they proved the platform's real advantage with a molecule called flap endonuclease 1, FEN1.
FEN1 is the perfect target for validation. It's a DNA processing enzyme, so its active site is basically a DNA magnet.
Which means it's fundamentally unsuited for DEL selections. The DNA tag would just confuse the enzyme.
Completely. You'd get massive false results. But SELs, they successfully discovered potent FEN1 inhibitors.
So that validates the entire barcode-free platform? It opens up these previously inaccessible target classes?
Exactly. The ones where the DNA tag was just too biased to get a clean read. This is a game changer, especially for academic groups who want to explore targets like transcription factors without needing that massive HTS infrastructure.
That's a powerful step. So SELs solve the problem for finding tiny molecules. But what about the really difficult targets, those, extended protein interfaces where you need a totally different kind of structure?
Right, let's pivot from rapid screening to rational design for those hard targets.
Yeah. We're talking about protein-protein interactions, or PPIs.
We are. And these interactions are the basis of nearly all biological signaling. The problem is the interfaces where these proteins connect are often shallow, they're broad, and extended.
They don't have those nice deep pockets that traditional small molecule drugs like to just slip into.
Exactly. So you need something bigger than a small molecule, but smaller than a full antibody. And that's where macrocyclic peptides or cyclic peptides come in.
The key is their cyclic nature, that ring structure.
Correct. It provides structural rigidity. And that rigidity does two crucial things. First, it gives them enhanced proteolytic stability.
Meaning they don't get chewed up by the body's enzymes instantly.
Right, which leads to much better pharmacokinetic behavior, a longer half-life. And second, their fixed shape allows them to engage those tricky, shallow PPI interfaces much more effectively.
The structural advantage is clear. And the sources focus on a fascinating and really critical target for these peptides, the SLAT2-ROBO1 signaling axis.
Yes.
Why is everyone so focused on blocking this pathway right now?
It's a major frontier in oncology. The SIT2-ROBO1 pathway is a key player in how tumors grow, and, more importantly, how they evade the immune system.
Okay.
So when this pathway is active, say in an aggressive cancer like glioblastoma, it leads to the buildup of these immunosuppressive cells called tumor-associated macrophages, or TAMs.
So the tumor is using this pathway to basically put the brakes on the immune system.
It's telling the immune system to stand down. So if you can disrupt this axis, you might be able to reprogram that tumor environment and turn the immune system back on.
And they used an advanced computational strategy to do this.
They did. They didn't just randomly test peptides. They used a fragment-based approach called DES3PI 2.0 to digitally map the high-density binding regions, the hotspots, on the protein interface.
And then the computer generated the ideal macrocyclic peptide sequences to fit those spots.
Exactly. It's a completely rational computer-guided design process.
So what came out of it?
Well, after synthesis and validation, they identified several SLIT2 targeting peptides. The lead candidate, which they called SP4, proved to be the most potent.
And they confirmed that with competitive biolayer interferometry.
Yes, with an ICU of 41 MEM, which is solid quantitative evidence that the computational design actually worked. It generated a molecule that blocks the biological interaction.
And the beauty of making it cyclic shows up in its developability profile, right? Because a lot of peptides just fall apart the second they hit human plasma.
This is really the holy grail for a lead candidate. SP4 showed tremendous stability. It had a half-life of 7.6 hours in simulated intestinal fluid.
Wow.
And even more tellingly, after an hour in human plasma, 89.4% of the molecule was still intact.
So the ring structure is providing crucial protection.
It's a huge green light for continued development. It shows that these computationally designed macrocyclic peptides are actually viable drug scaffolds.
So we've covered the flexible chemistry in the lab with SELs and this rational molecular design with macrocyclic peptides. Now let's jump to that third piece, the intelligence layer. The AI that predicts the very biological relationships these molecules are trying to target.
Right. We're moving to PLM-interact. So we've all heard of protein language models, PLMs, like ESM2. They're incredible at predicting the structure of a single protein from its amino acid sequence.
Okay.
But here's the problem. When it comes to protein interactions, traditional models are kind of blind because they aren't inherently aware of the interaction partner.
It's like they know how one sentence works, but they don't know how it relates to the sentence right after it.
That's the perfect analogy. They can analyze the structure of the word, but not the context of the conversation.
So what does PLM-interact do differently?
It overcomes this by extending and fine-tuning a pre-trained PLM-ESM2, in this case, to jointly encode protein pairs. It explicitly teaches the model to look at two proteins at the same time.
Sort of like the next sentence prediction task in natural language processing?
That's exactly what it's like. The transformer's attention mechanism learns the shared context and the relationships between those two proteins.
So that joint encoding gives the AI the ability to anticipate how two proteins will dance together. How did it perform?
Its performance is stunning, especially in cross-species prediction. They trained PLM-interact only on human PPI data.
Oh, okay, just human data.
But when they tested it on 5 evolutionarily divergent species, mouse, fly, worm, yeast, E. coli, it showed state-of-the-art results. For example, it had a 10% AUPR improvement on yeast data over the next best model.
And let's explain that. AUPR, area under the precision recall curve, it tells you how reliable your predictions are, especially with imbalanced data.
Which biological data always is.
So a 10% jump on yeast, when it was only trained on humans, that means the AI is learning the fundamental rules of protein language.
It's learning the universal grammar that applies across the tree of life, not just memorizing human biology.
It's unlocked something fundamental.
It seems so. And what's more, a fine-tuned version can predict the subtle impact of single mutations on that interaction strength, telling you if a mutation will make the binding stronger or weaker.
Which is invaluable for understanding genetic diseases. Give us an example of that.
It successfully predicted that the N151A missense mutation in frataxin, a protein linked to the severe neurodegenerative disease Friedreich's ataxia, that this mutation decreases its binding affinity with its partner, ISCU.
Wow.
For a family of diseases that are so hard to treat, having an AI that can model the molecular failure just from a change in sequence, that's a massive step forward for designing a therapeutic.
And the sources also highlight its relevance to, well, global health, infectious diseases.
Yes. PLM-interact showed an 11.9% improvement in the MCC score. That's another reliable measure in predicting virus-human PPIs.
And that capability is absolutely vital.
It is. It helps conventional virus host prediction tools gain molecular detail. That's critical for anticipating zoonotic events, for predicting the emergence of novel viruses, essentially giving you drug targets before a pandemic even starts.
This whole deep dive has been a rapid-fire tour. It really shows how the early drug discovery pipeline is being modernized on, multiple fronts at once.
We have fundamentally changed the workflow. We've moved from laborious bias screening like HGS and DELs to rapid, flexible, untagged screening with SELs.
In a lab.
In the lab. At the same time, we're rationally designing complex molecules like macrocyclic peptides for previously undruggable targets. And guiding all of this is the intelligence layer. AI tools like PLM-interact that predict the fundamental rules of biological engagement.
The convergence of these three things, the chemistry, the structure, and the intelligence, that's what the sources are calling the true revolution.
I think they're right.
And critically, they promise to democratize rapid early-stage drug discovery. By streamlining everything, these advanced techniques become accessible to a much wider array of researchers.
It moves the science out of just the mega-pharma facilities and into smaller labs all over the world. It's the promise of bringing down the cost and increasing the speed of that first most crucial step.
And that discussion about PLM-interact really suggests that the fundamental language of molecular biology, the protein sequences, is being learned by machines in a way we're only just beginning to leverage.
We really are just at the beginning.
So here's the final provocative thought for you, the listener, to mull over. If AI can now predict interaction based purely on sequence, what previously undruggable target, a cancer target, a neurodegenerative target, maybe a novel virus, what will be solved first when we combine this predictive power with the flexible chemistry of SELs and the structural sophistication of macrocyclic peptides?
The rules have changed. The race is on.
Thanks for diving deep with us. We'll see you next time.